Today in this class I am explain about gene transfer methods in plants. First one, vectors for gene transfer. Gene cloning deals with the transfer of the desirable gene into a host cell. The cell which receives desired gene is called host cell, and the desired gene cannot be directly introduced into a host cell because the desired gene does not carry the origin of replication. Hence, the desired gene has to be introduced into an autonomously replicating extra chromosomal bodies. These extra chromosomal bodies are present in bacteria, bacteriophages, and viruses. These extra chromosomal bodies transport the desirable DNA fragment into a host cell, and they are termed as cloning vector or cloning vehicle. The cloning vector may be plasmid or phage or virus. And the requirements for cloning vector. A cloning vector is a DNA molecule and it should have the following important features. First one, it should replicate independently of the chromosome of the host cell. It should replicate independently of the chromosome of the host cell. It should carry a marker gene that enables the selection of cells that acquire it. And third one, it should be easily isolatable from the host as a discrete molecule, and it should contain an assortment of unique restriction endonuclease cleavage sites, and it should occur in large number of copies. And next one, types of cloning vector. The gene cloning vectors are used to transfer the desired gene into the host cells. Hence, the vectors are also called gene transfer systems. The important gene transfer system are the following. First one, plasmids. Second one, bacteriophages. Third one, viruses. Fourth one, shuttle vector. And fifth one, expression vector. Sixth one, cosmids. And seventh one, transposons. First one, plasmid. Plasmids. Plasmids are the extra chromosomal self-replicating double-stranded and circular DNA molecules, and they are present in bacterial cell. Naturally occurring plasmid can be modified by in vitro techniques. Cohen in 1973 for the first time reported the cloning DNA by using plasmids as vector. For example, plasmid and PBR 322. And the types of plasmids. First one, relaxed plasmids or multi-copies plasmids, and stringent plasmids or single copy plasmids. Third one, conjugative plasmids. Fourth one, non-conjugation plasmids. types of plasmid. First one, relaxed plasmid. Second one, stringent plasmids. Third one, conjugative plasmids. And fourth one, non-conjugation plasmids. And first one, relaxed plasmids. The relaxed plasmids multiply continuously. So, a bacteria may contain 1000 plasmids at a time. Normally, each bacterial cell contains 10 to 20 copies of plasmids. These plasmids are ideal for gene cloning. And second one, stringent plasmids or single copy plasmid. These plasmids are present in less numbers. Sometimes a bacteria may contains only one copy of the plasmid. So these plasmids are known as single copy plasmids. And third one, conjugative plasmids. These type of plasmids carry a set of gene known as transfer gene or TRA gene. These genes promote conjugation. Each cell contains 1 to 3 copies of conjugative plasmids. They have high molecular weight. And non-conjugative plasmids. In this type of plasmids, the TRA genes are absent. Each bacterial cell contains large number of plasmids. They have low molecular weight. And characteristic features of an ideal plasmids vector. All the plasmids present in the bacterial cells are not useful for genetic engineering works because some plasmids lack the property of carrying the foreign DNA fragment along with their own sequences. Some plasmids replicate only in the cells from which they were isolated but fail to replicate in the host cell. A plasmid vector must have the following character features for the successful gene cloning. They are: the plasmid's DNA must be smaller in size. The plasmid DNA must be smaller in size, and the plasmid must have the capacity to transfer the foreign DNA fragment into the host cell, and it must be a shuttle vector which replicates both in the donor cell and in the recipient cell. And fourth one, it must integrate the foreign DNA fragment into the genome of the host cell. The plasmid must have one or more genetic markers to detect the presence of cloned genes in the organism. The plasmid should not have any pathogenic property, and the plasmids must have its own regulatory genes for the replication. And the properties of plasmids. Plasmids have the following characteristics. Plasmids are circular double-stranded DNA molecules. They are genetically dispensable elements in the sense that they are usually non-essential for the growth of the cells. They replicate independently or autonomously. They have an origin of replication naturally in them, and they are passed on to the daughter cells during cell division. And sixth one, they may carry very important genes for antibiotic resistance production, for antibody production, for degradation of a large number unusual substrates such as herbicides or industrial effluents, and genes for nitrogen fixation, and they propagate the DNA fragment linked to them by in vitro ligation, and they may be present in large number of copies, and they can acquire chromosomal gene by several mechanisms, and they rely on the DNA replication enzyme of the host cell for their replication. However, the initiation of replication is controlled by plasmid genes, and certain plasmids do not show any phenotypic traits. Such plasmids are called cryptic plasmids. And they have high transformation efficiency, and they have convenient selectable markers such as antibiotic resistant toxin production for transformants and recombinants. They have the ability to clone responsibly large pieces of DNA, say about 5 kilobase pairs. They are of low molecular weight, and they have single site of a large number of restriction enzymes, preferably in genes with a readily scorable phenotypes. They are easily isolated and purified. And plasmids type. Plasmids are duplex supercoiled DNA molecules. They range in size from 0.05 to 10% to the size of the bacterial chromosomes. The low molecular weight of plasmids has certain advantages, and the number of copies of plasmid in a cell is referred to as copy number. When there are one or two copies, the copy number is called low copy number. When there are one or more copies, that the copy number is called low copy number. When there are 20 or more copies per cell, the copy number is called high copy number. The plasmid PBr322 is a high copy number plasmid. This high copy number is an advantage in the sense that a receive mutation cannot bring out any changes, and plasmids can recognize into conjugative and non-conjugative, and this characterization of plasmid is based on the presence or absence of set of transfer gene called TRA genes. The presence of TRA genes promotes bacterial conjugation, and the plasmid can also be categorized based on the number of copies. Has first one low copy number plasmids can high copy number plasmids. The former is called the stringent plasmids, and the later as relaxed plasmids. The naturally occurring plasmids are classified into 5 types based on the main characteristics coded by the plasmid genes. It is grouped into 5 main types. First one, F plasmid, R plasmid, coal plasmids, degradative plasmid, and fifth one, virulence plasmids. First one, F plasmid. F plasmids are fertility plasmids. They have the ability to promote conjugal transfer of plasmids. These plasmids carry only transfer genes. These plasmids may denoted as F plus and F minus, which means those having the fertility factor and those without it. These plasmids are not used in gene cloning. And next one are plasmids. These carry genes conferring on the possessor resistance to one or more antibacterial agents such as chloramphenicol, ampicillin, tetracycline, and any metal, and are transfer resistance. And the plasmid RP4 found in Pseudomonas is an example of R plasmid. This R factor was discovered in Japan in 1955. The R factor is widespread in certain strains of almost all pathogenic bacteria. The plasmid genes often encode for enzymes that chemically inactivate the drug or by active export eliminate it from the cell. And third one, coal plasmid. These plasmids contain genes for the production of bacterial toxin known as colicin, which kills other bacteria. The bacterial toxin are generally called as bacteriocin. These bacteriocin are active only against closely related strains of bacteria. Toxins of this type that are liberated by strains of Escherichia coli called colicins. The colicins are simple proteins. Several different types of colicin have been isolated which kills sensitive cells by different mechanisms. And the plasmids containing genes for such toxic substances, colicins, are called coal plasmids. And depending upon the nature of colicins, there are different types of coal plasmids. They are coal B, coal E1, coal E2, coal I, and coal 5. The toxin for Pseudomonas is called phycocins. And fourth one, degradative plasmids. These plasmids, which have genes for enzymes that enable the bacterium to metabolize unusual substrates such as taurine, xylene, and salicylic acid. These plasmids are also called dissimilation plasmids. The plasmids type responsible for the ability of certain Pseudomonas species to break down difficult to degrade industrial solvents such as toluene and xylene. A combination of several plasmids when transferred to Pseudomonas allow the bacteria to break down complex hydrocarbons and other compounds present in crude oil. And the bacteria containing these plasmids have a potential use for treatment of environments contaminated with oil spills. And the last one is virulence plasmid. These plasmids have genes that confer pathogenicity on the host bacterium. For example, Thai plasmids found in Agrobacterium tumefaciens induce crown gall disease on dicotyledonous plants. That's about the plasmids. Thank you.